Hello, I'm Dmitry Popkovsky, Chief Scientific Officer of Fluxel Biosciences. In this short tutorial, I would like to describe for you just how easy it is to use Fluxel's MitoXpress Intra assay to measure cellular oxygen concentration. MitoXpress Intra is a nanoparticle-based cell-penetrating probe formatted for use on a time-resolved fluorescent plate reader to study cell metabolism under controlled oxygen environment. MitoXpress Intra is a perfect complement for other Fluxel's microplate assays, including MitoXpress Extra to measure oxygen consumption rate and pH Extra to measure extracellular acidification rate. All of Fluxel's assays come complete with a detailed product specification sheet. When you unpack the MitoXpress Intra product containing a vial of the nanoparticle-based MitoXpress Intra probe, you can put it straight in a refrigerator for later use. To conduct the MitoXpress Intra assay, you will additionally need the following: a cell culture hood, cell culture incubator, time-resolved fluorescence plate reader, black body clear bottom tissue culture treated 96 or 384 well plates, and pre-made recommended respiration media. In this tutorial, we're plating A549 cells in a 96 well format at 60,000 cells per well. We recommend that you plate cells in the inner wells marked white and fill the outer wells marked black with PBS or growth media. Two of these outer wells, H11 and H12, as shown, are used as blanks during the measurement. Now return the plated cells to the cell culture incubator for overnight incubation. On the first day of the MitoXpress Intra assay, you will pipette 11 mL of your cell culture media into a 15 ml plastic tube and warm this to 37 degrees centigrade in a water bath. You will use this solution to reconstitute the MitoXpress probe. Remove a package of MitoXpress Intra probe from the refrigerator and bring this along with the pre-warmed culture media into the cell culture hood. Reconstitute the probe material at the bottom of the MitoXpress Intra vial using 1 ml of the pre-warmed culture media. Aspirate 4 to 5 times gently and then transfer this 1 ml to the remaining 10 ml of pre-warmed culture media to create your 11 ml of probe stock. Remove spent culture media from the plated cells, taking care not to dislodge cells from the base of the wells. Next, using a multi-channel or repeater pipette, add 100 microliters of the 11 ml intraprobe stock to each well, taking care again not to dislodge cells. Return the plate to the cell culture incubator for overnight incubation. On the second day of the MitoXpress Intra assay, you will pipette approximately 38 ml of your pre-prepared respiration media into a 50 ml plastic tube and warm this to 37 degrees centigrade. Using an aspirator, remove the spent intraprobe from the plated cells, taking care not to dislodge the cells from the base of the wells. Next, using a multi-channel or repeater pipette, add 100 microliters of the pre-warmed respiration media to each well, again taking care not to dislodge cells. We recommend that you perform a wash step to remove any residual intraprobe from the well by simply repeating the previous aspiration of spent media and addition of 100 microliters of the pre-warmed respiration media to each well. Lastly, remove the spent respiration media and this time add 150 microliters of the pre-warmed respiration media to each well. Remember to additionally add respiration media to the wells H11 and H12, which will be used as your blanks. You are now ready to quantify the cellular oxygen concentration in your assay plate. Test compounds may also be added or treatments applied to each well at this time, if so desired. In this particular instance, we're using the Fluostar Omega offered by BMG LabTech. The instrument should be turned on in advance to allow for temperature equilibration. This plate reader has been programmed to the correct measurement parameters. The plate is measured kinetically, typically for 30 to 90 minutes, but can be measured for longer. Drug treatments can be applied during the measurement by pausing the instrument, withdrawing the plate, making compound additions, and then resuming the measurements. In this particular case, the BMG LabTech data analysis software will automatically calculate the lifetime values for the MitoXpress Intra probe. The initial part of the graph shows basal oxygen levels which correspond to respiration of the resting cells. When the treatments were applied at around 10 minutes, we see a transient process and then a new respiration state after about 30 minutes. The response is presented in lifetime scale, but it can also be converted into oxygen scale using the calibration function. In this second experimental example, we're recording the cellular oxygen concentration in A549 cells using MitoXpress Intra. Oxygen concentration is controlled by an atmospheric control unit fitted on the Fluostar Omega reader and stepped down as the experiment progresses. Note the dramatic difference between applied oxygen concentration and the actual concentration at the cell monolayer. When oxygen in the solution measures 8%, the monolayer concentration is really at only 3% due to active cell metabolism and local oxygen depletion. Then, when oxygen in the solution measures below 3%, in this experiment the A549 cells are effectively anoxic. MitoXpress Intra is therefore a valuable tool for any studies requiring the measurement of cellular oxygen concentration at the monolayer, cell in suspension, or 3D cultures, and where drug effects are studied under controlled and physiological oxygen levels. Thank you for watching. This and also other technical information can be found on Luxel's website www.luxel.com.